Hello everyone, today we are going to continue our topic Salmonella, which is belonging to the family Enterobacteriaceae. I am Dr. Sharad Deshmukh from S.S. Jaiswal College, Arjun Morgaon, District Gondia. So let us continue over the earlier topic. So now we will see about the biochemical reactions of the Salmonella. As far as the biochemical reaction is concerned, first we will see about the fermentation of the various sugars by the Salmonella species. So they are able to ferment the glucose, mannitol, maltose, and they fermenting with the formation of the acid and gas. But here the exception is the S. typhi, which ferment this sugar with the acid production only, while the other species, particularly the paratyphi species, they ferment this sugar with the production of acid as well as the gas, but the S. typhi ferment with the acid production only. They are the sucrose negative, that is they are not able to ferment the sucrose. One more concept is they are non-lactose fermenting, we have already seen. So they are able to ferment the glucose, mannitol, maltose, but they are not able to ferment the sucrose and the lactose one. The most important character, they are non-lactose fermenter. Now coming to the other biochemical reaction, as far as the indole test is concerned, they are negative, then they are MR positive, they are VP negative, they are citrate positive, except the Salmonella typhi, which may vary, and generally they show the negative citrate test. While the urease test is negative, which is one of the important tests to differentiate them from the other species, particularly the Proteus species, where the Proteus are the urease positive but the Salmonella are the urease negative. And most important test for this is H2S production test, which is the positive for the Salmonella species except Salmonella paratyphi A, which is the H2S negative. We have already seen in the biochemy, in the culture character, the S paratyphi A show the green colonies on Wilson Blair because they are the H2S negative, while the other species they are the H2S positive. That is most important character in the identification of the Salmonella species. Now let us come to the next aspect about Salmonella. We will see about the antigenic structure of the Salmonella. Here they possess the three types of the antigens. So three antigens are identified. The first one is the Flagellar antigen, what we refer as the H antigen. Second one is the Somatic Antigen, that what we will say that is the O antigen. And third one, which is antigen present in the Salmonella, is the surface antigen, that is also referred as the VI antigen. So in this case we will see one by one about each antigen. First coming to the H antigen, that is the Flagellar antigen, that is present on the flagella. So this antigen which is present on the flagella, and these are the heat labile protein. So it is the protein in nature and it can be destroyed by the heat as well as the alcohol. So but the most important character is it is strongly immunogenic. Among all the antigens, the Flagellar antigen is strongly immunogenic and they produces the loose clumps with the antisera. That means when this antigen is mixed with the antibody against the Flagellar H antigen, that is anti-H antibody, that will form a loose fluffy clump which resembles the cotton wool type of the agglutination is seen. That is a characteristic feature is seen in case of the Salmonella. Next one, H antigen is dual in nature and it is occurring in the two phases, that is they are occurring in phase 1 and occurring in the phase 2. So they occur in the two phases, the phase 1 antigen and the phase 2 antigen. Here the phase 1 is the species specific. So this antigen is present in the particular species. That means if it is present in the S. typhi, then it may not be present in the S. paratyphi A. So phase 1 antigen is the very species specific, while the phase 2 antigen, it may be shared by the many species of the Salmonella. So it may be present in more than one species. It can be shared by the Salmonella species. So this is the phase 1 and the phase 2 antigen, that is dual nature, which is seen in case of the Flagellar antigen. Next we'll come to the next antigen, that is the somatic antigen, also known as the O antigen. This O antigen is a very complex structure. So, and it is present in the cell wall of the bacteria, that is the Salmonella species, and it is the phospholipid, protein, polysaccharide complex, and it is the heat stable. While the earlier one, the H antigen, it was the heat labile, and this one is the heat stable, and it is also resistant to the alcohol and the acid. Now in this case, the most important character, it is the very less antigenic as compared to that of the H antigen, where we have already mentioned in the H antigen they are highly antigenic, and this O antigen is the less antigenic, and O agglutination occurs very slowly and it occurs very at high temperature, that is at 56 degrees centigrade, while that of the H antigen that reacts very fastly and that takes place at the 37 degrees centigrade. And in this case also, when this antigen is mixed with the antisera, that will form a specific type of the agglutination, and that is the granular type of the clumps are seen, which are the compact and chalky. That is distinctly seen when you add the antisera to the antigen. And this antigen is not the single factor but a mosaic of the many antigenic factors, and about 65 different O antigens have been recognized, and they are also designated as the 1, 2, 3 like one. So this antigen characteristically you can differentiate from the H antigen, as the agglutination is occurring slowly, it is less antigenic, it is the heat stable, while the H antigen is the heat labile, it is strongly antigenic, and agglutination takes place rapidly at the 37 degrees centigrade, and that is the cotton wool type seen in the H antigen. Here it is seen as a compact and chalk powder like the agglutination is seen. That can be differentiated between the somatic antigen and the H antigen. Then coming to the third type of the antigen, what we referred as the VI antigen, or it is also referred as the surface antigen. So as we have mentioned, it is the surface antigen, that means it is present on the surface of the bacterial cell, and it was first described by the Felix and the Pitt. This antigen is present in the particularly Salmonella typhi, S. paratyphi and S. dublin. This species carries this type of the surface antigen, and this VI antigen, it is also the heat labile, and the bacilli which are agglutinable with the O antiserum at normal temperature but become agglutinable if boiled for 1 hour. That means here we have mentioned bacilli which are inagglutinable with the O antiserum as such if they possess the VI type of the antigen, because this VI antigen is completely covering the O antigen which is present on the cell wall at the normal temperature. But if you boil this culture, then this will get destroyed and that become agglutinable with the O antiserum because of the destruction of the surface antigen. In this case, the one more character is it is contributing to the virulence. That's why they are mentioned as the VI antigen responsible for the virulence. They act as a virulence factor because of the inhibiting the phagocytosis, and they act as the virulence factor. So it is the less immunogenic, that is another important character, and very low titer of antibody is produced against this antigen. So earlier we have seen the H antigen that is strongly immunogenic, O antigen is comparatively less immunogenic, but it is very less immunogenic as compared to the earlier 2 antigen. And the presence of the VI antibody that indicate the carrier state of an individual. So if it is detected in the sera of a patient, that indicate that the person is in the carrier state, while the other two antigens are important for the diagnostic purpose, that is O antigen and the H antigen. The V antigen is lost due to the repeated subculture. As we have already mentioned, it is present on the surface, and also it is the purely immunogenic, and it can be lost when it is repeatedly subculture on the media, and that may lose this V antigen, and the strain will become agglutinable with the O antiserum. So this was about the today's lecture.